Okay, we're going to start off taking a look at gel electrophoresis, and hopefully this is something that you did in cell biology, so it's not completely unfamiliar to you. You will be doing it again in this class. And so this picture just shows a gel apparatus. There's a gel in there. It's in a big box with some buffer, and we're going to run electric current through the gel to separate molecules, right? So the purpose of gel electrophoresis is to separate molecules based on size. The basic principle is you're using electricity to pull molecules through the gel. Smaller molecules are going to be able to wiggle through the gel faster than bigger molecules. So in a given amount of time that you run the current through the gel, it's like a race, and the small molecules go farthest and the bigger molecules stay behind. So if this is a finished gel at the end of gel electrophoresis, and this was a DNA gel, DNA has a negative charge, so it is pulled towards the positive electrode. So you would have loaded your samples in the back here and then turned on the current, and the DNA molecules are getting pulled through this gel, and so whichever little collections of molecules are closest to the positive electrode, these would be smaller molecules, so fewer base pairs. Remember, we measured DNA by length of base pairs, so these molecules have fewer base pairs in length than the ones back here. And each of these bands represents many, many copies of a molecule. This, on the left-hand side, this is a size ladder, so often in your gel you will run a ladder. You buy it from a supply company, and it has pieces of DNA in it already pre-cut, and the sizes are known. And so if you run that with your samples, you don't know how long your different fragments are, but you know these, so you can line up and you can say, okay, well, these bands here, these molecules that got this far, they are just a little bit smaller than this known member of my ladder here. So that gives you an idea of the size of the things in your gel. Okay, so a little review of how you would do gel electrophoresis. You're going to prepare the gel, right? It's a buffer solution, some salt and water, plus the agarose. Agarose is a polysaccharide from seaweed. It's a thickener, so that's what gives the gel consistency. You pour the gel into a gel tray. So this is a gel tray with no tape on the end here, but normally you would have tape. And then you put a comb into the gel while it's still liquid, so that as it hardens up, this comb is going to create little pockets called wells in the gel that you can add your samples to, right? And then you're going to prepare your samples. So if we're doing a DNA prep, you need to add some loading buffer to the DNA. The loading buffer will have a molecule that's very dense, so that when you put your samples in these pockets, in these wells, because these little holes are going to be filled with liquid, and your DNA samples are going to be in a liquid. If you put liquid in a liquid, it's just going to float away. So you have to add some loading buffer to mix your sample with something heavy, so that when you add your liquid samples to these liquid pockets, these liquid wells, your samples will sink down into the well. Also, you want to have dye, right? So you've got dye in your loading buffer, because when you're running the current through and your DNA molecules are getting pulled through the gel, you want to see approximately where they are in the gel, so you don't run them off the end, which can happen. All right, so here is a person loading their gel. So we use micropipetters, and they're loading into those little fluid-filled pockets. They're loading their DNA samples, and this is kind of a cool picture of a close-up of some wells. So you can see these little shadowy rectangles that you put your samples into, and then the blue here is showing that loading buffer mixed in with the DNA and how it kind of sinks down into the well. And then here is a gel apparatus again. So the wells are up here, right? And you take your gel and you put it in this gel box. You put buffer in it, so salt water all around it, so it'll conduct electricity. You hook up the leads to your power supply, and the red is the positively charged electrode. So we've got the red down here, got the black, the negatively charged electrode, and that is going to cause the DNA molecules up here to pull through the gel towards the positive electrode. Right? So when you set up your gel, you put it in that big chamber we were just looking at, you add the buffer, you load your wells, and then you run the gel. So you connect up your leads, and you make sure you put that negative electrode near the wells because you want the positive electrode far away so that the DNA will run to the positive electrode. Biologists say that DNA runs to red, right? R for run, R for red. Then when you're done running your gel, you need to stain your gel. So the stains in the loading buffer are just so you can watch the overall progress of your samples through the gel. They just give you an approximate location of your samples. They don't give you the actual bands. And so after you're finished, you stain. One stain that is sometimes used is ethidium bromide. Ethidium bromide sticks to DNA and it fluoresces under UV light. So now this gel's been put on a UV light and you can see all those bands glowing. One downside of using ethidium bromide is it sticks to DNA. And you can imagine that things that stick to DNA are not very good for you. As a matter of fact, they are carcinogenic. So you can use, ethidium bromide works great. Small amounts of it gives you great glow. You'll really see your bands. But because it's toxic, sometimes it's not used in teaching labs. And if you do use it, you have to be careful. Gloves, eye shields because you're using UV. So it's something to be careful with. So you stain the gel, you view the gel. As I said, there's different stains that are possible. And then you note the positions of the bands. The bands are the stripes, and each band represents a little collection of DNA molecules that had the same size. And I already mentioned you can use the ladder to estimate the size of your bands. Okay, so hopefully that was review from cell biology. You'll get a chance to practice this in lab. The other technique I want to mention that's important for DNA sequencing and genome work is the polymerase chain reaction. And actually, I don't think I could say too much about the importance of the polymerase chain reaction in biology. The polymerase chain reaction enables us to make many, many copies of a small amount of DNA, and it has revolutionized biology in so many ways. It's the basis for forensics, when they collect samples at crime scenes and there's only a little bit of a sample and you want to study it. You need polymerase chain reaction to make copies of whatever DNA you've got so that you have enough to work with. And it's just an amazing tool, and everybody uses it practically. It's such a valuable tool. So the purpose of polymerase chain reaction is to make many copies of a section of DNA. The basic principle is that you're doing repeated cycles of DNA replication. So you're just copying and copying and copying. So you make copies, then you make copies of the copies, right? And so what you get is this rapid expansion. And the thing that makes sense to me, but most of you are too young to remember this, is there was a shampoo commercial in the 80s. And I actually have a link to it because I know you're too young to know it, most of you.
When I first tried fabricate organic and so on, and so on, and so on. And believe me, there's still nothing like the original fabricate organic. Gives you super shine, super body. Super fresh smelling hair. Try it, and you'll tell your friends about it. And they'll tell their friends, and so on, and so on. Fabricate organic shampoo and conditioner.
Okay, that's enough of the shampoo commercial. It's not the shampoo, but it's the I'll tell two friends and they'll tell two friends and so on, and that screen filling with her. That's how PCR works. You make copies, then copies of the copies, and so on and so on. And so if we look at the process of PCR, the way that it works is you have your DNA, right? So in this DNA somewhere, there is a sequence that I want to copy. And the first thing I'm going to do, if I want to do DNA replication, I have to separate the parent strands. And in the lab, we do this with heat. So you apply heat, and that's going to denature the DNA. And I know you know denaturing of protein means unfolding of the protein, right? And that happens because you break the weak bonds. So when we talk about denaturing DNA, we're not really talking about unfolding the DNA so much, but we are talking about breaking the weak bonds, the hydrogen bonds, that hold the two strands together so that they will separate, right? So step one is denaturing the DNA, separating those strands. Step two, you need to provide a 3' end for DNA polymerase to work on. Remember, DNA polymerase can't start a chain on its own. So in a cell, primase comes along and builds a primer, which gives that 3' OH. Well, in the lab, what you do is you add primers. So you have lots and lots of primers that have a sequence that you have chosen because you know that sequence is flanking, it's on either side of the DNA you want to copy. And you put lots and lots of primer in the mix. And so then you let your DNA cool a little bit so that the hydrogen bonds can come together again, and your primers are going to be complementary to the sequence that's around the DNA you want to copy. And when you cool this down, you let those primers stick. Step 3, elongation. Now, DNA polymerase has what it needs. It has a template to copy, it has a primer to get started on, and of course you've put lots of nucleotides in the mix, so it has the raw materials. So DNA polymerase, this little green circle here, jumps on those primers and starts extending them out, elongating the DNA chain. So where originally we had one piece of DNA, now after one round of PCR, we have two pieces we've copied, okay? And each of our copies starts where that primer bound, right? So the 3 basic steps. We use heat to denature the DNA, breaking those hydrogen bonds, and typically that's heating 92 to 94 degrees. Then we're going to cool so that those primers can stick, and the fancy word for sticking is annealing. So the primers anneal, and again you have to have lots and lots of primers in that mix, and the primers provide that 3' OH that DNA polymerase needs. The sequence of the primers is complementary to the DNA that's around the DNA that you want to copy, and you want to have lots and lots of primer in your mix because you don't want those original strands that you separated with heat, you don't want them to come back and stick together. You want the primers to stick to those open chains. So you put way more primer in your mix than you could possibly need, so that the most likely thing to stick to your separated DNA molecules are primers. Right? And typically you're going to cool to about 45 to 55 to let those primers stick. Then you want to raise the heat again to allow elongation. And the reason that you're raising the heat is because the DNA polymerase that's used to do PCR is actually a DNA polymerase from a thermophilic bacterium called Thermus aquaticus. And so you want to bring the temperature up to its optimum. And its optimum, we call the enzyme Taq polymerase. So the bacterium is Thermus aquaticus. So Taq is for the name of the bacterium, and then polymerase because it's a DNA polymerase, right? And it likes temperatures about 75 to 80 degrees, so you raise the temperature up to that. Taq polymerase does its job, jumps on those primers, and starts building DNA strands complementary to the original. Before scientists figured out about using Taq polymerase instead of sort of a mesophilic DNA polymerase, every time you did PCR, after every reaction, you had to add more enzyme because you kept denaturing your enzyme. And then somebody got the bright idea to use Taq polymerase, and now the whole process is automated and it's fast and it's great. Right? So after that, you just repeat steps 1 through 3, right? You told 2 friends, now you want them to tell 2 friends, and so on. So you just keep going through these same 3 cycles: heat, cool, heat. Right? And so if we look at this picture, we were here, we were to step 3, right? Then you do it again. You heat to denature, so we're going to separate our strands. Cool to allow the primers to stick. Raise the temperature to elongate. Now we've gone from 2 to 4 strands. Do it again, and we've got 8, right? Do it again, and we've got 16. And very, very quickly, faster than you would imagine, you have lots and lots of copies. This is what a thermocycler looks like, right? Where you put your samples inside. It's got a little block that has space for these teeny tiny little tubes. And this picture is just showing you again these cycles of temperature and what's happening during these cycles of temperature. This building of the copies of your DNA, right? And so after just 30 rounds, doing those steps 1, 2, and 3 30 times, you can have over 1 billion copies of your DNA. So basically, you run your PCR reaction overnight, and the next day you've got plenty of DNA to work with. Okay, the machine is called a thermocycler, or also called a PCR machine. It automatically cycles through the temperatures. So You prep your samples, stick them in, set your timing, and push the button. And 30 cycles will give you over 1 billion copies, right? So you just run it overnight. Okay, so that is gel electrophoresis and PCR. Last clip up next, I'm going to talk about